Hello and welcome to the presentation of my poster entitled Properties of Re-fermented Green and Roasted Coffee Beans. The aim of this research was to investigate the effect of re-fermentation with Bacterium Bacillus subtilis and yeast Vicar Hamomyces anomalus on phenolics content and antioxidant properties of coffee beans. We used Coffea arabica green beans as our basic material. The beans went through a process of soaking and sterilization, inoculation, fermentation, and drying. We prepared fermentation control, which went through the same process without inoculation step, and we also investigated raw and processed material. All samples were roasted, and extracts were prepared from green and roasted samples. We used Folin method to evaluate total phenols content, and ABTS method and method based on autooxidation of linoleic acid to investigate antioxidant activity. Here we can see results for the fermentation process. Both selected microorganisms grew under given conditions. However, in case of Bacillus, the growth was over 10 times greater. In the previous studies, where Vicar Hamomyces anomalus was also used, a greater growth was reported, however only after 24 hours. We can speculate that during our fermentation there was a growth peak, and after that the number of living cells started to decrease. We also noted some significant changes in the pH value of fermentation mix. They might be due to metabolism of microorganisms, but also due to activity of coffee enzymes during the soaking step. Here on the left side we can see that the amount of phenols in the re-fermented green coffee beans was significantly greater than in fermentation control sample. It might be due to enzyme activity of microorganisms, specifically proteolytic or pectinolytic ones, which might have hydrolyzed some complex phenols in coffee matrix and made them easier to extract. In case of antioxidant activity on the right side, we can see that the fermentation caused the decrease of activity in case of inhibition of linoleic acid oxidation compared to fermentation control and unprocessed samples. In case of ABTS method, we can however see that yeast fermented green coffee beans had greater antioxidant activity compared to unprocessed sample. Roasting caused the increase in all investigated properties, although fermentation control and unprocessed samples still exhibited greater values than the fermented ones. There are some exceptions in case of ABTS method results, however full statistical examination was not possible. In previous studies where fermentation of green coffee beans with different yeasts was investigated, opposite trend was noted. There was increase in both total phenols content and antioxidant activity of roasted re-fermented coffee beans. The effect of roast seen here is not uncommon. During this process, melanoidins are created and they can compensate for the loss of natural antioxidants of coffee. In case of re-fermented coffee beans, we can also speculate that there were some changes in basic coffee constituents, such as proteins or carbohydrates, and that might have had the effect on the compounds created during roasting step. In conclusions, we can say that there is a possibility to change coffee properties with additional re-fermentation step. There is, however, a need for further studies regarding not only the choice of microorganisms, but also to understand the changes in basic coffee components and their connection not only to antioxidant activity, but also a sensory quality of final product. Thank you for your attention.